Okay. I made a little tiny URL thing. If you want, all of our slides are available in a Google Drive that we will at some point share with you, as well as copies of the supplemental data. But just if you want, my slides aren't very complicated, so you don't really need to follow along in them, but there's a little URL you can go to if you want to follow along in them. I loved Yulia's presentation, and most notably, her picture of those two crossing paths and the point at which the functional data intersect with the anatomical data is the best roadmap I've ever seen. So thank you. And then a little start song. I was like, oh, I want to analyze my data. My little roadmap is much less pretty. I'm starting here with data acquisition. And you acquire some functional data. And these are they're potato chipped on purpose because your functional data are distorted. And then the thing I'm focusing on is this thing I've been playing with lately, which is during that scanning session, also acquiring a T1 weighted EPI. At first, Camille Uludog shared with us a work in progress, and that was when we had an older scanner, and that died with the older scanner. And then on our newer scanner, we were lucky enough to have, it's a CMRR, right? We have resources everywhere. And Eddie Auerbach was like, sure, I can put a T1 into your regular old EPI. So now we just have like a little tick box, which we're like, yeah, put a inversion pulse with some kind of a specified inversion time. But I'll talk a little bit more about that in a second. But this was a special pulse sequence that we had to create for the moment after I tried 6 other things and fell flat on my face. I seriously spent a year trying to figure out how to add adequate T1 weighting to my own EPIs, and it completely failed until I read Camille Uludog's paper and other people created this pulse sequence for me. And then anatomy, and this is one of my weaknesses. I have a weaknesses slide coming up, and we have a lot of discussion going on about what do you use for your anatomical reference scan. And so far, all we've talked about is defining depth in the gray matter in the space of this anatomical reference scan. I'll say one more thing about that in a second. There's an alternative, which you could define, and Renzo does, right? Your cortical depth in the space of your functional data. And so skipping all of these questions about did I correctly align my functional data to my anatomical data, do I know where I am? You can skip all those questions if you could just define the cortical depth in the space of the functional data. I can't yet. This T1 weighted EPI is a great step in that direction, and I think I have in my hands the tools I would need to do that and completely skip this anatomical reference scan, which I really believe would be the superior pipeline. Just stay in your distorted space. You know you're distorted. Don't try to correct them. Live with them. Work there. So I should add that to my list. It's on my list of weaknesses. Anyway, but here's what I'm doing. Is I've got my work functional scans. I've got these T1 weighted scans that I'll show you that really help me know where I'm at. And then I've got some kind of an anatomical reference. I actually have 2 anatomical references. In this scanning session, I acquire one that I use as a go-between for a different 3T acquired MP Rage. It's like the 1990s called and they want their data back. I've got them. Because you can just walk up to a scanner, turnkey, put a person in, get an MP Rage, feed it into FreeSurfer. You don't know anything about what's happening in the box in FreeSurfer, and outside the other side comes a really adequate looking segmentation. I'm not a teaching professor, I'm a research professor. I have an academic appointment at an R1 research institution in the United States, which means my role statement is 50% research, 30% teaching, 20% service. But I tend to take that seriously because I absolutely love my undergraduate teaching commitments, and public engagement is the reason that I actually get up and come back to my job every day. So I'm only a 50% researcher, and much to the dismay of some of the powers that be around here. Oops, I don't spend 100% of my time researching. And so these weaknesses that I'm showing you are because I've got a lot of demands on my time. One of them is I need to train undergraduates how to get through this pipeline, and it's got to be a lot of turnkey solutions. So I'm showing you a lot of turnkey solutions, partly because of philosophy, partly because there's only one Cheryl and she's got to do all of these jobs. Did that make sense? Like the data I'm working with, they're limited and they're not the best. That's why we're here. We're all learning from each other. My processing pipeline looks like within scan motion compensation on the data. And I do my motion compensation before I do my distortion compensation. I've tried it both ways. I haven't seen that it has a big impact on my data. And it philosophically sits with me better to do motion correction before I do warp field estimation or distortion compensation. So I do within scan, and then since I have this handy dandy distortion matched T1 weighted scan, it is actually my reference. Everything is going to move into the space of this forward phase encode T1 weighted EPI scan. So I do within scan motion compensation for all my functional data, and then I do a between scan discovery to find out what rigid body 12 parameter rotation and translation would I need to move to correct for subject motion throughout the scan using my base scan as the forward phase encode T1 weighted EPI. I do some math to actually extract from the data a T1 map, which has really lovely gray matter, white matter contrast, which is what we're always going for in life. Then I feed those into the algorithm. I'm using AFNI's Q-warp. Same thing as FSL's top-up, same as Brain Voyager's top-up, probably forward and reverse phase encode scans to discover the truth in the middle and estimate a warp field. And then 3D and warp apply is happy to combine my motion and distortion compensation parameters with my warp field estimation to produce nominally distortion and motion compensated functional data, to which I apply some kind of general linear model to discover statistical parameters that represent how the brain works. And that's the candy-colored brain mess up there. And then in parallel, my anatomical data have been moved, but not warped, just rigidly rotated into my functional space, another improvement I could make. And from them, I extract depth labels. Either I use an equivolume solution on top of the FreeSurfer segmentation. That's what I've done for years. This year, I've finally been getting brave enough to try Laney. I need to work out some bugs there. But I get some kind of depth labels. I always use rainbows to represent depths. And so combine your statistical map with your depth labels gives you parameter profiles. So there's the big overview of the map. Here in words, didn't animate those, is the things I use to do it. Renzo.
Audio would work best if you're in front of the computer.
I don't like standing by my computer. That's true. All right, we'll be over here so the sound's better. AFNI's 3D Volreg, AFNI's 3D Q-warp, AFNI's 3D Alineate. AFNI loves saying 3D, so I do my within scan on Volreg. I do my unwarping with Q-warp. I use nwarp apply to apply. I use 3D Alineate to discover the transformation from the anatomical to the functional space, and then either FreeSurfer or Laney to get some depth labels in there. So that's the pipeline I'm sharing, and I'm talking now very, I lack Julia's patience to give you step-by-step screenshot guides, so today I'm just giving you kind of overview, and tomorrow we're gonna hack through it if you want to. So here's me articulating my current worries and then my current weaknesses. You can read them as well as I. I do distortion compensation. Is my distortion compensation good enough? Am I actually compensating for distortion? Where's my ground truth after I compensate the distortion in my functional data? Is it actually shaped like my anatomical data? I guess I want you to look at the data files that I have shared and come to your own conclusions about that. I'm in the process of writing up this pipeline just as a methods paper, so it's documented out there somewhere, and so I can get another line on my CV. And I'm playing around with comparing, because I could also, I do in this scanning session, and I did share with you in the data set that I shared, there also is a reverse phase encode functional data set. My hypothesis is that the superior contrast of the T1 map will produce superior outcomes from this warp field estimation when you do distortion compensation. But that's an empirical question. And so in my hands are also reverse phase encode functional data, where the gray matter, white matter contrast is much weaker. I hypothesize that that provides less information for the Q-warp algorithm to figure out where the truth in between is, because it can find the edges of the brain just fine. It can find alignment between high contrast features just fine, but for like shifting around signal where the contrast is low, why would any algorithm succeed when there isn't a visible gray-white boundary? So that's the hypothesis. That's why I'm using this T1 weighted EPI to guide distortion compensation instead of the regular old functional data. Is it a meaningful difference? I don't know yet. It was fun to try, and so that's what I'm sharing, but it might not mean that much. And neither is doing a perfect job. And how do we know if our functional data ended up in the right place when we put them down on top of our anatomical data? So that's why the title of my contributions to this workshop is like distortion compensation and registration, because that's where I spend my life worrying. And we'll talk more about my weaknesses in a second. But moving into this question of manual editing of segmentations, I'll show you places where it really would matter if I were analyzing that part of the brain. But with in the work I'm doing, I started out, when I was a grad student, I was using Brain Voyager, and I did lots of manual segmentations, and all of my brains were blue. And then something would happen, and I would have to reacquire a data set or reprocess something, and I would have to redo my manual segmentations. And my life is too short, and there are so many other wonderful things to do with it, that manual segmentation was just one thing I didn't want to spend my time doing. And that's been kind of a guiding principle for me. And maybe it's sheer laziness, I don't know. But I've done everything I can to avoid manual segmentations of the anatomical data. Is that going to kill my career and make my results invalid for the rest of my life? I'm starting to wonder when I see all this really gorgeous treatment of anatomical data that everyone else is presenting here. So that's another worry of mine. And along the same lines, you can see all my worries are really quite anatomical. Because it provides a turnkey, reliable solution, I still use MP Rages off a 3T scanner for which the out-of-the-box FreeSurfer was optimized. And yeah, I would love to see that. At the break, we were talking about MP Rage versus MP2 Rage versus multi-echo MP Rage. Could we get all of those in the same head and then apply all of these segmentation algorithms to them and see how, because even just the contrast with which you acquired your data is going to determine exactly where you find your gray matter, white matter boundary. So I think we really just struggle as a field to be really sure about what depth is. Okay. Those are my worries and my known weaknesses. I've said it already, so we don't need to belabor it, but my data are a little bit archaic for the reasons I've mentioned. And the thing I should be doing is segmenting my T1 weighted EPIs. Why am I not doing that? Maybe I will be 2 months from now. We'll see. And then my registration algorithm. To my understanding, boundary-based registration algorithms are far superior to like just the full image contrasts weighting that I'm using when I call AFNI's 3D Delineate. Really, since I have these gorgeous T1 weighted EPI scans, I really should be using a nonlinear warping of my anatomical data to my functional data at a minimum, using a boundary-based registration algorithm. So, belying my ignorance, there are users in this room who are more familiar with AFNI than I. Does AFNI have a functional, high-quality, boundary-based registration algorithm built into 3D Delineate? Does anyone know? I'm not seeing any head motion. I haven't been able to find one, and if it did, I would use it in a heartbeat. So everyone get on the phone to NIH, hint, hint, and bring us a boundary-based registration algorithm in AFNI. Otherwise, I can pipeline it out into a different software. But to answer these questions about why do you use AFNI as opposed to other ones, exquisite documentation, so very scriptable, really accessible. You know exactly what's happening to your data at every step. And so I'm kind of attached that way. Okay. So a brief summary of the data that I've shared. And I will tonight, I promise, apply my pipeline to this standard data as well by tomorrow. The hotel Wi-Fi would not work for me. Why is Cheryl staying in a hotel if she's local? The floors in my house are being redone right now, and so I can't go home. And so I stayed at the conference hotel, and the Wi-Fi sucks. But tonight I will apply my analysis to the pipeline to see if I get answers anything like Renzo's. So what I'm sharing is 0.6 millimeter isotropic 2D gradient echo EPI. I'm a vision scientist. I always acquire my data in coronal slices, always just aiming for the back of the head. I should have brought my coffee mug. I have a coffee mug that has my retinotopy printed on it with a little picture of exactly what the slice should look like if you're going to put it in my head and hit my 3 degrees eccentricity representation in primary visual cortex, because that's where all my stimuli are. So I'm like, chug the coffee, put the mug on the shelf, and be like, grad students, aim here, I'm getting in the scanner. So that's the slice. So I'll see a lot of coronal slices, which I sometimes remember to show like a little axial and a little sagittal section so you can see what a really tiny bit of the brain I am covering. But we can get a 2-second TR. Everybody knows that there's multi-slice, multi-band acquisition in the world. The bands, like the narrowness with which you can specify the separate RF pulse that excites the different slices for simultaneous the different slabs for simultaneous multi-band excitation, those slabs can't be skinnier than about 3-4 centimeters. So when you're doing super high-res work with really skinny slices and you're kind of limited to how many slices you can get per slab, we can't use multi-band acceleration because it just sends our G factors through the ceiling, because we end up with cross-excitation between the bands, and it kills your signal-to-noise ratio. Hence the really limited coverage, even though it seems like I should be able to multiply that times 4 by turning on multi-band 4. I've got 32 receive coils. You can't. Ixnay on that first line, I was going to share the 4 task runs, but we're sharing them by uploading them to a Google Drive, and that just made the data set so huge that I stripped them out and I'm just sharing the localizers. But if you love this data set and want more, talk to me. I'll hook you up with some task runs. So I've got 3 localizer runs. This is a relatively short data set, so it's a little bit amazing that with 0.6 millimeter isotropic resolution and just 3 4-minute scans, we can get pretty decent activation. And then the matching reverse phase encode run in the regular old EPI, and then these distortion and position matched T1 weighted EPIs. So if you're not at all familiar with this, then my brief explanation will not be everything you need. But in a nutshell, I've got 25 slices, and I've grabbed 5 of them to illustrate. So across the columns, 3 of them to illustrate. Across the rows are 3 different slices, so you'll recognize 3 different shapes in the 3 different rows. And then across the columns are 5 different time points out of the 55 that I acquired, which was overkill. But so here's a time series of the signal intensity in 3 voxels, 1 in each of those 3 slices, color-coded. So focusing on the top row, that slice, there's a red circle centered on a voxel that I picked in CSF. And over the course of the scan, the intensity in that voxel had this pattern. Why? Because the way these T1 weighted EPI acquisitions work is there's an inversion pulse, but then it takes you 2 seconds to acquire all of your slices in some particular order. So the first one has a very short inversion time, the last one has that short inversion time plus 2 seconds. And then on your next excitation, you swap the slice order, and it's swapped each time. And so the effective inversion time for each slice changes throughout the scanning session. I'm so glad somebody invented this. So over the course of time, each slice sees a different inversion time and gives me a different intensity. And then to process that, I can go back into the headers of the DICOM and pull out the slice order information and the specific inversion times for each slice on each TR, and then do some math and estimate a T1 value for each voxel based on its particular intensity profile as it's cycled through these different inversion times over the course of my acquisition. So for the yellow voxel, I picked something that was, I don't even know what, also looks kind of CSF-ish, and for this blue voxel, I picked something that was more in the tissue, and you can see that they had different, it's complicated. I can't just read those and tell you what the T1 was, but they each have different temporal profiles for the intensity because of the different inversion times. Does that make any sense at all? Okay. I love that pulse sequence. I could just do it all day. That's the time during which I watch my princess movies. because we have a Disney Plus subscription here, and so when I'm the subject, I'm like, oh, let's do 60, 70 TRs, I got time, and then I watch all the stuff that I can't watch at home for my anatomies and my T1-weighted EPIs. Okay, this anatomy, Natalia Petridou's group published a paper, I don't know, 10, 15 years ago, setting out some TR and TI parameters that were ideal for optimizing the contrast for the stria of Gennari, which should be a nice ground truth for us. Those are those input layers that I was complaining they walk up and down as you move through the curvature of the brain, and they're deeper than you would think. So during that same scanning session, we get this anatomy at pretty high resolution, so we can try to see the stria of Gennari. And at the end of the day, when I do my laminar analysis, I should see a peak in layer 4 where of just the T1 intensity profile. Truth be told, it doesn't work that well, but I only have one of these and the signal noise ratio is really low. But this is one of my ground truths, is you can image the stria of Gennari. If you're lucky enough to work in a primary sensory area, you're going to get this bright band through the middle of cortex that helps you ground truth your depth definitions. And then from a different scanning session, an MP-RAGE. All words, yay. So I've shared this pipeline, and as I went to share it, I realized I was going to have to share more than is probably useful, but I'll show you what it is. So I relied mainly on AFNI. So to use this code, we're going to hook you up with a server. That server has AFNI on it. If you're going to do this on your own server later or your own machine, you're going to have AFNI installed on that machine. I'm not too picky about versions, really. Like I'll check as versions change if anything changed about my analyses, but I'm pretty flexible in thinking about versions. There's a part in there where my scripts are going to call MATLAB and launch MATLAB so it can run Nordic. So show of hands, who knows already what Nordic is? All right, keep that hand up if you've used Nordic already. Okay, so we're like half and half. So for those who have not yet witnessed the magic trick of Nordic, noise reduction, what's the rest stand for? Independent components. By independent, yeah. Steen works some magic. And the idea is that we are pushing the limits of resolution, which means our voxels are getting smaller and smaller, and as your voxels get smaller and smaller, you move from the physiological noise-dominated regime to the thermal noise-dominated regime. So down at 3T with 3 millimeter voxels, you've got all the signal-to-noise ratio in the world you want. Your biggest problem is your participants' respiration, motion, cognitive fluctuations, physiological noise. We can push, as we push to smaller voxels, the crossover point at 7T is somewhere around a millimeter. Your physiological noise is in the noise, and the dominant source of noise. It's still there, it's still a problem, but your biggest noise source is now just thermal noise, random independent fluctuations in the electronics that you're using in your hardware. And key to that idea is that it's independent. And so if you analyze the structure of the data, you can find components that look independent. I have no idea how the algorithm does it. Other people can teach you Nordic, not me. And that's what Steen did when he wrote this Nordic algorithm. So it takes your data and it looks at the noise structure, identifies the part that is independent and explained by nothing else in the data, and then strips it out. It's got to be Gaussian, I don't know. And it seems like that could go really wrong in many ways. And sometimes it does, so don't look at your data. But my gosh, if it doesn't work. And I'm like this independent observer. I didn't develop the algorithm. I'm not like paid by the folks using the algorithm. But it is game changing. My data just have twice the signal noise ratio they used to after they come back out of Nordic. And I cannot see any evidence for spatial smoothing. I cannot see any structure to what's changed in the data. I just have twice the signal to noise ratio after I run Nordic over my data than before. Okay, off my soapbox. Love it. So worth it. So we launched MATLAB to run Nordic. That gave me no end of grief moving this pipeline to a new server, but I got it sorted. So we're MATLAB dependent specifically because of Nordic. Has anyone put a memo into Steen that we want him to rewrite that into Python? After the workshop, we'll talk to Steen. And then I'll use either FreeSurfer or Laney to define. So those are my external other people's software dependencies. And then there's this thing called MinPy, and I started writing it maybe, I don't know how long ago. Time flies. It was when I decided my lab needed to switch from using MATLAB to Python. For some reason, I decided to write pipelining software, because Python is super easy to call from the command line. It's just much lighter weight than MATLAB. I was like, okay, here's my application. Command line tools for simplifying the commands that my students need to learn when they're going to process data in my lab. So it used to be named CAO, my initials, Cow Pie. But that was just embarrassing and awkward. And then I had this dream that maybe all the vision research labs in Minnesota, we would start sharing and co-developing this pipeline. So I named it MinPy for Minnesota. And if that ever happens, it's not going to be this year. So I had this like awkwardly named MinPy as if it's like the Minnesota pipeline. It's not. It's just what I wrote to handle data. And so I shared it in the software directory, and it is a work in progress. It is always a work in progress. It was written as my Python learning experience, but it gets the job done. So I've shared it because the easiest way for me to share my scripts, because all my data analyses are always scripted. What happens before I publish every paper is I wipe all the analyses, and then I run one batch script that regenerates all of the results. And those would better match the last version of figures regenerated. But I really have this commitment to just automated, no user decisions, no manual interventions. Can a pipeline run it? So I had the process.sh script for the paper that's about to go out the door on the data sets that I'm sharing with you. I hope it's about to go out the door. And I just like cleaned it up to share, and then I realized, oh my gosh, all it is calling MinPy. So now I'm sharing MinPy with you. It might be too much of a hassle. As MinPy runs, each step also logs all of the calls that it composed to AFNI. But like in 3 flags to MinPy, you can generate an AFNI command that has like 12 flags. And so it just saves us a lot of typing. So that's the ecosystem that I've shared, all wrapped by this process.sh script. And I thought that there was a little picture, nope, of the study directory, but that comes later. Okay, so that's the software. So I've told you about the data I'm sharing. I've told you about the process I'm sharing. And so now we'll just look a little bit at each of the steps as an overview. So as I wrote this to truth is I found a bug in how I was unwarping the T1 weighted EPI about 6 months ago. So I had to wipe everything, break it gently to the graduate students. It's okay, we're gonna reprocess all of your data before your conference, but we'll make it. And so we were tearing through it, and we broke it into chunks. So there's a pre-processed chunk, a depth files chunk, a spatial ACF chunk, a GLMs chunk, and then an ROIs chunk, which is just very last step, figure out where we are to extract some voxels to make laminar profiles. So pre-processing involves applying Nordic to the raw data to create an original data type, doing motion correction to create a MoCo data type, which is never actually used, but holds the important motion parameters you need. Processing the T1 weighted EPIs, extracting the warp fields from them, and then combining those warp fields with distortion compensation to create an unwarped data type. So that's the end of the pre-processing, is I have unwarped functional data. And then it aligns the functional data to the anatomical data. So we call that pre-processing. And when I say aligns, I'm careful about the order in which I put those 2 words. We align the anatomical data to the functional data. So the functional data never move except for being motion compensated and unwarped. Depth files, either I went to FreeSurfer and asked it for 20 equivolume. It's always 20, 21, how do you count the top and the bottom, but 20-ish equivolume surfaces, and then made a map, translated those surfaces back into volume space, so I had like 20 different volumes showing just the voxels, the ribbon of voxels that would intersect with each of those surfaces, and then just averaged them all together to create a single volume that has a floating point number in it for a relative depth from 0 to 20. And I never scaled it back from 0 to 1, so that's super awkward. It goes from 0 to 20, but we don't care. Or I fed, good, Renzo's distracted, don't let him hear the rest of this. No, don't listen. Don't listen. Just the entire ribbon file of the entire brain, because I couldn't figure out how to automate the patch making in a good way to get the RIM files I needed for Laney. We'll talk. Like, I know it can be done, but I couldn't figure out how to automate it right. I'm getting closer. So I just fed the whole ribbon file for the whole brain into Laney and added like a RIM on the top and bottom. And then it gave me back answers. And that's the current implementation of Laney. Are we going to fix it before tomorrow?
Sure, I didn't really get the problem. You want the entire brain, like FreeSurfer output converted to Laney. No.
Right, and we just took the whole brain mask and fed it into Laney, and Laney was perfectly happy to work on that and give us back depths everywhere. Except the problem we have is the way we formed a RIM for the top and bottom. We're underpopulating the very deepest and the very
that's
most.
An option, equal bins. I talk about it. That matches where you basically make sure that each layer has the same number of voxels, if that's what you want.
Yes.
Yes. Like you basically sort all the voxels based on the metric and then not like chunk it into bins of cortical depth that are normalized based on the depth estimate, but you basically do a sorting of all the voxels and chunk it into bins based on the number of voxels.
Okay.
If that's what you want. It's not geometry.
There's the thing, because equivolume conceptually would give you that answer. I felt like I must be doing something wrong if equivolume didn't naturally give me that answer.
Depends. Like the brain is ball. It's more convex than concave, right?
Net. It's got a bigger outside than an inside. Oh. Complicated, as always. Okay. I have to go teach during your... Oh, I teach this super awesome class where I take undergraduates over to middle school, and we like... never mind. I've got to go do that this afternoon. So I'm going to sit in on Renzo's teaching session tomorrow. Okay, so that's how I create my depth files. Okay, this is something that I really love, the spatial ACF. So AFNI has this 3D local ACF function that maps the local spatial autocorrelation functions for voxels. And I've never been able to, it's difficult to get the fMRI community to agree on what is an appropriate measure for smoothness in your data. Like we acquire at this nominal resolution of 0.6 millimeters. But we know that the actual, like two voxels next to each other are not independent of each other. There's some kind of smoothing or blurring going on in my data. And quantifying that, what is the true resolution of my data? Sure, I could chunk my data into 0.6 millimeter, but I could cut those in half and have a nominal image resolution of whatever I wanted. But the true intrinsic resolution, my ability to separate signals that are X millimeters away from each other, I really struggle with understanding how to measure that. And what I love is this local ACF function for estimating, fitting a Gaussian plus an exponential tail to a, but our data are so thermal noise limited that the spatial autocorrelation function is fundamentally Gaussian. And I love looking at that number to inform my sense of the true resolution of my data. But I would love some vibrant discussion on that topic as well, because just because I acquired my data at 0.6 does not mean that my resolution is 0.6. A conversation to have. But it takes forever because you have to run a GLM and then run a really complicated math on the residuals from that GLM. So that's a very time-consuming step that I usually leave toward the end. Then general linear models are quick. Since I'm not sharing the task data, that'll just be one basic GLM, takes no time. And then ROI making, we'll talk about later if you care. So the way MinPy organizes the data is that you have all your raw data over in a DICOM directory, and then there's a func subdirectory, and then down in there's a raw data type before Nordic, an original data type after Nordic, and then a MoCo data type, and then an unwarped data type, and as many other data types as you want to make and name as you try different analyses, and it's very flexible about how you name those things. Any files related to the reference anatomy will be in a volume directory. ROIs and surface-based files go in different places. I collect all my depth information in one place. And then there's an experiment directory where the raw behavioral data are stored with the DICOM, but the stimulus timing files that you need for your GLM and your GLM instructions are down in an experiment directory. So when you finally get access to this data set, it'll be all populated with all of these, and analyses will be done, if the rest of today goes well. Or if you want to repeat, just do it all from scratch, it'll take a bit of time, but the only things you need are the raw data, a Python file that's marked up to indicate which scans should be used, how, that guides all of the analysis for MinPy, And then the software that I've shared, as well as the GLN instructions and the 3T anatomy that's already FreeSurfered and SUMA'd from a separate scanning session. So that's the roadmap to the data. Okay, I'm just going to end with things to watch out for. So here I'm going to flip back and forth between the T1 weighted EPI, the map, the T1 map. These aren't perfect estimates of T1 parameters, but we expect to see a shorter T1 in the gray matter and a longer T1 in the gray matter and the longest yet T1 in the cerebrospinal fluid. So qualitatively, that is an adequate T1 map. This is the functional data as they came off the scanner, and it certainly is easier to see anatomical structure in the T1 map as opposed to the data. And I'm also quite happy to see that my claim that they are in the same position and have the same distortion, whatever the distortion in there is, I would say that those images are pretty well on top of each other, but not identical. And so the places in which they're not identical intrigue me most. This is coronal section, the back of the brain, so the sagittal sinus is here. This is my enemy. Hate the sagittal sinus. The amount of deoxygenated hemoglobin really perturbs the magnetic field. It really makes the inferior parts of the occipital lobe swing back and forth when it's distorted. And you're also seeing a lot of signal dropout because these are T2 star weighted acquisitions. And so I don't know that I have any business doing analysis of functional data down here. So I tend to put my stimuli down in the lower quadrant of the visual field, because the lower quadrant of the visual field is represented on the upper banks of the calcarine sulcus, and I move my stimulus and my analysis away from this mess. So accurate analyses of visual processes at this kind of resolution in the upper visual field in primary visual cortex, I'm not entirely sure it's possible with T2 star weighted imaging. So that's one of the things I debate with myself. Another thing you want to look out for is the results of my attempts at registering after I've applied motion compensation to my functional data and I put them down on top of my anatomical data. The bright blue and green lines represent the surfaces extracted from the anatomical data. I will say, I've no manual segmentation. I'm Cheryl. But you see a goober here. This is where the segmentation, for whatever reason, I didn't have a T2 weighted image as part of this. You can refine the peel surface segmentation with the T2, and for whatever reason on this anatomy, I didn't even include that. So here the segmentation has failed and is grabbing part of the sagittal sinus itself. So not only is my signal dropping out, but my segmentation is failing down here. So this is, a person should not draw any conclusions about any analyses that are done here. How do you automatically detect those places and leave them out? I don't know. So then here's me flopping back and forth. And here there's a slight shift between the two images because I failed to align them in the Google slideshow, not because there's an actual shift between the two, because you can see those blue lines moving. But again, I flip back and forth between these things for hours, wondering where do I think that my alignment between my EPI and anatomical data look great? Where do I have some serious problems? Where do I have some minor problems? And how good is good enough to move forward with laminar analyses? I don't know how to answer that question for myself in an automatic, unbiased way, but that's one of the quests that I continue to pursue. And then if I overlay some activation map on it, is that activation in the gray matter? If you have something go wrong in your path that introduces wicked spatial correlations, a quirk of the data set that I'm sharing is that it was acquired on the parallel transmit system on our 7T-AS. When y'all acquire data this afternoon, you're doing it on this single channel system, right, with a NOVA? Yeah. If you do it on PTX, to support Nordic's estimation of the uncorrelated noise in your data, you acquire 5 empty, you turn off the RF and you just acquire a bunch of noise volumes at the end, and you do it long enough that residual signal from excitation can just evaporate. And you take that last volume as a pure noise sample to inform Nordic's estimation of the noise. On the PTX system, for whatever reason, that doesn't work. There's some residual RF. We're not sure what's not getting turned off, but if you feed that last noise scan into the algorithm, Nordic discovers this structure, and your signal to noise ratio goes like through the roof. You've never seen such gorgeous data. but then your activation like blurs all the way through your slice volume. So you look for things like that as hallmarks, as something has gone horribly wrong in my data analysis. And so if this had been Nordicked the wrong way with a not true noise sample, then you would see big blobs of activation instead of just the occasional false positive showing up in the white matter. And every time I look at an image like this, I just pause and reflect over the fundamental problem that we are doing functional MRI with T2 star weighted images. Because, hey, look, here's a big vein. All of our good signal is coming from big veins. I'll talk later about some attempts I'm doing currently to devein things or just to keep analyses away from regions where you know you have a huge vein. But if just T2 star weighted fMRI is I don't know. Should we do it? So just think about that as you look at the data. And then at the end of the day, you take some region of interest and you take your depth labels. What's going on here? That's a just great question. They're not all lined up so you can't see. There's just some honkin' huge something going wrong right here, and just like, whoa, don't analyze here. And what do you do about that? How do you, if that's where your stimulus was and you need to know what the laminar profile is right here, I don't know what your plan is. Thank goodness I put my stimuli up. Well, that's not much better. So at the end of the day, you pick your region of interest, you think you don't have any problems there, you pull out your depth labels and you pull out your statistic representing what happened. What I'm showing here is even more complicated than that. This was an orientation tuning experiment. The participants saw 8 different orientations, and so every single voxel gets an orientation tuning curve fit to the response of that voxel to the orientations. We were seeing if at 0.6 millimeters we could get small enough to actually detect orientation columns, which are a canonical structure in human visual cortex. The answer is no. This is an orientation selectivity index that I'm plotting. I don't know whose pulse sequence I'm about to erase. Is anybody married to this pulse sequence? It looks fancy. If orientation is my x-axis and just response is my y-axis, and a neuron or a voxel has some response to any old orientation and then a preferred response, that's what you expect as an orientation tuning function out of primary visual cortex. If a voxel is super selective, it'll give you a super sharp peak, and this has low circular variance. Circular variance is a concept I really struggle with. Not conceptually, but just practically speaking. I always get it backwards. But you have low circular variance if you only have responses at one particular orientation, and then you have higher circular variance if your responses are more distributed around all the possible angles. And then the typical vision neuroscientist is going to compute orientation selectivity index as 1 minus the circular variance. So that's my y-axis, and so what we expect from neurons is that the inputs in the middle layers are relatively orientation non-selective. They're slightly selective, but then lateral inhibition refines the orientation selectivity. So the right answer for this experiment I dream of working at an institution where every marker on every board is always working. The right answer here is a U-shaped function, where the input layers have low selectivity, and then the deep and superficial layers, due to cortical processing, have high selectivity. That's the ground truth. We know this is true. I was trying to recover the ground truth of this experiment to convince myself that I'm doing science. We saw hopeful tips up, and if viewed with the eyes of faith, you could maybe see a U-shaped function. There is no U-shaped function there. Further consideration of this data set showed me that the process of fitting circular variance to the data is sensitive to noise such that you will just spontaneously get, by the quirks of how it fits and responds to noise, we will spontaneously get lower circular variance, higher OSI numbers when the data are noisier. So we did a scrambled control, and sure enough, just the noise scrambled control looks just like our data, where there's a slight peak in the deepest layers and then it comes down. And this is because the F statistics in our data are lower in the deeper layers, because the T2 star weighted functional MRI signal is a bit weaker in deeper layers for reasons that we're all still a little bit not sure about. But they are, and I don't like the most probable answer. And so the shape, any shape I see here, and some things do pass t-tests, and I do have a higher orientation selectivity in deeper layers than in middle layers. Completely an artifact of the noise structure of my data. So that's good. I did science, but really not the kind of science that I wanted to do. I have utterly failed to discover any kind of orientation selectivity in this data set. So I'm not even sharing with you the regressors for the orientation or all the task data, because it's going to be a challenge to get that paper out the door. But the reason I have these up here is using my slightly impoverished, brand new, bugs need to be worked out implementation of Laney versus my other not perfect process for defining depth using FreeSurfer. Are those two pairs of laminar profiles the same or not? I would argue that I would draw the same conclusions from both of those profiles, even though they have slightly different shapes. They would end up leading me to the same conclusion, which I consider to be a good and somewhat reassuring thing. But I will just end with this last note that is linked in the slide. So Farouk took a different subject out of this data set and did some Laney magic on it. So if you want to watch beauty and an entirely different way of segmenting gray matter and white matter using a gradient computation that had never occurred to me before, about a month ago or two, there's some gorgeous treatment of data. I don't even think I have a thank you slide. That's it. This is what I got. So I'll end here.